What is the carnitine shuttle and why does it exist? The carnitine shuttle, as the name suggests, involves carnitine. And carnitine is a molecule that helps bring in long chain fats from the cytosol into the mitochondria. So we're not talking about short chain fats or medium chain fats. These are long chain fats. And we want them to enter the mitochondria because that's where the process of fatty acid beta oxidation happens. Fatty acid beta oxidation is what helps break down these long chain fats into shorter chain fats, and it helps generate electron carriers, which end up going on to form ATP. And you might be thinking, well, why is the carnitine shuttle needed only for these long chain fats and not for the medium or short chains? Well, think about it this way. If you're moving a very large object into your house, you're probably going to want some help carrying it in. So you might need to hire some movers, and those movers are the carnitine. In contrast, the medium and short chain fatty acids, those are short enough, small enough that they can be brought in on their own. So they don't need their own separate helper system like the long chain fats do. So the carnitine shuttle, again, the point of it is to bring in these long chain fats into the mitochondria where they can undergo beta oxidation. So the first thing to understand with carnitine is that the cell actually has to take it up from the bloodstream into the cell. And this is via something known as the carnitine transporter. I'm just going to label it as CT. Once the carnitine is in the cell, it can then be conjugated to a fatty acyl-CoA. And when this reaction happens, when you get a fatty acyl-CoA and a carnitine together, you'll end up with an acylcarnitine. Now, the most common long chain fatty acid is known as palmitate. It is a 16 carbon fatty acid, also denoted as C16. It's 14 or longer, which classifies it as a long chain fatty acid. Now, the enzyme that conjugates the carnitine with the palmitate is known as carnitine palmitoyl transferase 1 or CPT1. So we have our CPT1 transferring the carnitine onto the palmitate, forming an acylcarnitine, a fatty acylcarnitine. So this molecule looks like this. It's a fatty acyl group, and now it's linked to a carnitine. Okay, so now what? First of all this is taking place in the cytosol. We need to eventually move into the mitochondria. So there's an enzyme that's going to translocate the acylcarnitine that we see here, the fatty acylcarnitine, across the inner mitochondrial membrane. And that enzyme is known as carnitine acylcarnitine translocase. It's exchanging an acylcarnitine going into the mitochondria. We're going to draw the mitochondria over here for just a regular old carnitine, one of these circles here coming out of the mitochondria, where it can be reused. Now, once the fatty acylcarnitine is actually in the mitochondria, it needs to be converted back to the fatty acyl-CoA form so that it can enter the beta oxidation cycle. So here we have an enzyme that is taking our acylcarnitine and it's popping off the carnitine, and in its place, it's reattaching that CoA. And so the enzyme that does this is essentially the opposite of CPT1. It's known as CPT2, carnitine palmitoyl transferase 2. And now this fatty acyl-CoA can enter the beta oxidation cycle. Okay, so now what happens to the acylcarnitine profile when we have a defect in each of these enzymes? We've got 4 enzymes here to walk through. Let's start with carnitine transporter. Carnitine transporter plays a very important role in the reabsorption of carnitine from the renal tubules back into the body. The idea here is that carnitine is a small molecule that gets filtered into the renal tubules, and it has to be reabsorbed in order to re-enter the body. If you lack this carnitine transporter, well, guess what? You're not going to be able to reabsorb that carnitine. So your carnitine levels in your urine are going to be high, and the carnitine levels in your blood are going to be low. Free carnitine is also known as C0. So when we send an acylcarnitine profile on a patient with carnitine transporter deficiency, we're going to have low C0. That is the hallmark of carnitine transporter deficiency, is a low C0. That carnitine is gone. It is in the urine. It is no longer in the blood. Let's move on to CPT1. What happens if we can't conjugate carnitine to the fatty acyl CoA? What's going to happen to the carnitine levels in the blood? Well, they're going to be high because there's nothing for it to conjugate to. There's no problem reuptaking the carnitine into the body. The issue is actually conjugating the carnitine with the fatty acyl CoA. And so your levels of C0 are going to be high. And if your levels of free carnitine are high because you can't make any of these acylcarnitines, well, guess what? All of those long-chain acylcarnitine species are going to be low because, again, CPT1 is involved in conjugating carnitine to the long-chain fats. You are left with carnitine, and you cannot make the long-chain fatty acylcarnitines that you would want. So that's why you end up with this particular pattern of elevated C0, i.e. free carnitine, and low long-chain acylcarnitines. Okay, let's move to CACT. If we can make this acylcarnitine, but the problem is transporting it from the cytosol into the mitochondria, what do we expect? Well, we should expect elevated levels of this long-chain acylcarnitine species. So we're going to have elevated long-chain acylcarnitines, especially C16. We're also going to have other long-chain species that rely on the carnitine shuttle that are also going to be elevated, again, because they cannot enter the mitochondria where they would be further broken down. We're just going to write here long-chain. We're talking about the long-chain acylcarnitines here. Because you're using up the carnitine to make the fatty acylcarnitine, the free carnitine levels, the C0, is going to be low. So it is exactly the opposite of what we see in CPT1. CPT1, the C0 is high and the long-chain acylcarnitines are low. In CACT, the C0 is low and the long-chain acylcarnitines are high. Let's take a look now at CPT2. If we can bring in the fatty acylcarnitine into the mitochondria, we just can't convert that final step from the fatty acylcarnitine into the fatty acyl CoA. Well, we're going to have the same problem that we did here with CACT. The C0, this free carnitine, can't be released. Carnitine is still going to be stuck to the acylcarnitine species. Therefore, the C0 is going to be low. Similarly, you're going to have lots of these long-chain fatty acylcarnitine species that are stuck in the mitochondria. So you're going to have elevated long-chain acylcarnitines, which could be C14, C16, C18. To summarize, we've walked through the carnitine shuttle. The purpose of this shuttle is to assist long-chain fats to get into the mitochondria. There's a series of 4 enzymes that are very important for this process to take place: the carnitine transporter, CPT1, CACT, and CPT2. And for each of these, it is only going to be the long-chain acylcarnitine species that are going to be disrupted, not the medium or the short-chain species. In addition, you can have disturbances of free carnitine. It can either be high, as in the case of CPT1, or low, as in the case of CACT or CPT2. The treatment for these disorders, we'll walk through them in another color. For carnitine transporter deficiency, the treatment here actually is to give carnitine. That's going to help increase the levels of free carnitine in the blood. So there's free carnitine and the bound carnitine. The bound carnitine is the acyl carnitine. It's bound to that fatty acid, so it's no longer free. That's what I mean when I say free carnitine. Let's move to CPT1. The issue is that we cannot take these fatty acyl CoAs and convert them into acylcarnitines. One solution is to reduce the amount of fatty acids in your diet. That is the mainstay of treatment for CPT1 deficiency. You want to limit the amount of dietary fat, palmitate these long-chain fats, which are very prevalent in the diet. We want to limit those because patients cannot use those fatty acids to make cellular energy. They are stuck as fatty acyl CoAs. They cannot be made into acylcarnitines. And if you have too many of these fatty acyl-CoAs hanging around, these can be toxic. So you certainly want to limit the amount of these fatty acyl-CoAs that are in the body. The way you do that is with a low-fat diet. In addition to the low-fat diet, you can actually give something called triheptanoin or C7. Patients can also get medium chain triglycerides. So medium chain, not long chain, medium chain triglycerides. And why is that? These shorter fatty acids don't have any problem getting into the mitochondria by themselves. They do not rely on this carnitine shuttle whatsoever. So ideally, we want to provide the body with a source of energy that it can use. We don't want to provide it with something that it can't use, that it's going to get stuck on, as is the case with these long chain fats. So by giving these medium chain fatty acids, you allow the body to undergo beta oxidation and to use the byproducts of that to make ATP. And it turns out that you can use these medium chain oils also for CACT and CPT2 deficiency. The principle is the same. Because you cannot bring in these long chain fatty acids to the mitochondria, instead you bypass it and you give medium chain fatty acids, which the body has no problem bringing in to the mitochondria and which you can use to make energy from. Now, let's take a look at CACT. So in addition to restricting dietary fat and giving medium chain triglycerides or triheptanoin, we can also give carnitine because we expect the C0, the free carnitine, to be low. So the general principle is if something is low in the blood and you can supplement it, then you want to go ahead and do that to raise the levels of whatever it is that is low. If you ever have a patient with low carnitine levels, you want to give them carnitine supplementation to raise their carnitine levels into a normal range. And the same treatments apply for CPT2. So we want to give carnitine because it's low, and we want to restrict the thing that is building up. So low-fat diet. Patients can also get medium chain triglycerides or triheptanoin. That's the mainstay of treatment for CPT2. Now, one of the other hallmarks on labs that you can see in a patient with a fatty acid oxidation disorder is something known as hypoketotic hypoglycemia, low ketones and low glucose. And this tends to happen whenever the patient is sick. So when they've been ill and they're not eating, their blood sugar is going to be low. Usually the body's response is to make ketones, which serve as an alternative source of fuel. So if you have trouble undergoing fatty acid beta oxidation because you have a long chain fatty acid oxidation disorder, guess what? You're going to end up with not only low glucose, but also low ketones. And this is one of the hallmarks of a fatty acid oxidation disorder. So if you see hypoketotic hypoglycemia, you really want to be thinking about a fatty acid oxidation disorder. We'll put a link in the description for the clinical features associated with each of these disorders, but in general, they can affect the organs that tend to rely on fatty acid oxidation the most for energy. And these include the heart, so you can get a cardiomyopathy, the skeletal muscle, so you can end up with rhabdo, which is muscle breakdown. And as with many other metabolic disorders, the liver can also be affected. So some patients can present with liver failure. So that was a summary of the carnitine shuttle and the disorders that affect it. I hope you found this helpful. If you did, feel free to give this video a like and consider subscribing to our channel and our newsletter where you can help support our work. Thank you.